Good afternoon. I think we are going to start our seminar today. My name is Josefa Pepa Antón. I'm professor of microbiology from the University of Alicante in Spain, and I'm the head of the group of molecular microbial ecology. We work with microbes, including viruses, of different types of environments, mainly hypersaline environments, and also different marine systems, including sediments and corals and marine plants. We work with the holobionts. And our main focus today will be on the viruses in hypersaline environments. So I'm going to start with the presentation, and we have. So let me. Okay. So I hope you see the presentation well. And as I say, we will talk about viruses in hypersaline environments. So first of all, a kind of disclaimer. So this is working. Why are we talking about viruses in a astrobiology seminar? Okay, I'm not an astrobiologist, as I must admit, but I think, and I'm not the only one who thinks so, that viruses are a key piece to try to improve our understanding of life here in the Earth and in any other possible place. Viruses are an essential part of the biological systems. And here you have a couple of reviews that address these aspects. And I think they were very timely because they stress the need to include viruses in the astrobiology schemas. We need viruses have like, well, as you know, there is a discussion on whether viruses are alive or not. Strictly speaking, they are not alive, but still they are a life signature. And besides, viruses may have genomes of any type of nucleic acid, and this makes them very interesting to try and to address other types of life. So I think viruses are an essential part of life, and they have to be taken into account when we want to study life here or elsewhere. So, as I said, I will talk about viruses in hypersaline environments. As you said, as you know, hypersaline environments are systems which have a very high salt concentration. How high? Well, the standard definition is that a system is considered to be hypersaline whether it has at least 2 times the salt concentration of seawater. The salt concentration of seawater, as you know, is like around 3.5%. Depends. Mediterranean can be a bit higher, depends. And I will talk mainly about the very high salt systems, those that are close to salt saturation, that is around 30% sodium chloride, but can be higher because there is like over saturation. And some hypersaline environments have different types of extremes. They are not only extremes because they have salt, but they may have a high pressure, they can be acidic, or they can have a very high concentration of what are called the chaotropic salts that induce the disorder in the biomolecules. Here you have some examples of very high salt systems. These, as I say, approaching salt saturation. They like Tyrrell in Australia, the Mono Lake in California, which is also high pH, salters. I will focus on salters. and we also have, like in Atacama, here on the right, we have soil, salty soils, rocks, which are inhabited by microbes and their viruses, and these are extremely rough because the water concentration is very low. We also have like more common environments, you say, like domestic environments, like this salted cod. This is a picture from a supermarket. You see this pink color is not like natural, it's because the cod got contaminated with haloarchaea. You can find halophilic microbes in basically in all the domains of life, okay? And within bacteria, archaea, and eukarya. And you see here, and this is a bit of decoration, but this is a tree of the whole living beings, and then you have archaea here on the right and bacteria here, and you have a very wide distribution of halophilic microbes. Halophilic microbes are those that need salt, and when they are extremely halophilic, they need like more than 50% salt. And here you have some pictures. When you in different environments with high salt, you can find different, as I said, organisms from different domains, from the three domains, like this algae Dunaliella, Artemia, it's an animal, and then you have yeast, even plants, and a lot of different cyanobacteria and these microbial mats. But we are going to focus in like the highest salinity along the salinity gradient, as I said. We're going to focus on the most saline systems within the hypersaline environments. And we work mainly with these solar salter systems. Solar salt, this is just a model system, a model salter, but it's very useful to explain the basics on the microbiome of these systems. You have here, this is a picture from a salter here in Alicante, and you have here at the bottom the Mediterranean Sea, and this red arrow is the water that goes through a series of ponds. And let's follow this yellow green arrow. The water goes through different ponds, and it goes through a sequence, a serial precipitation of salts. Depending on the solubility, different salts precipitate, and at the end, you have these pink saturated brines. Okay, so you have a physicochemical gradient due to the salt concentration and the salt composition, and this is accompanied, as I would say, by a biological gradient. And this is a very extreme system at the end with, I said, salt over the salt saturation and quite widespread. Those are coastal solar salters, but there are other types of salters. But the interesting thing that I wanted to show you, and this is why we work with these systems, is what you see here. You see that you go from marine salinity to saturation, to salinity above 35%. This is very high, okay? And these changes along the chemical gradient go with changes in the biological component. You have prokaryotes that increase in normal coastal water. You may have like 10 to the 6 or maybe more, it depends. But at the end of the gradient, when you go to the last ponds, which are called crystallizers, you have like 10 millions, 100 millions of cells per milliliter. The prokaryotic diversity decreases in these systems, but the microdiversity, the intraspecific diversity is still very high. Archaea, this is very relevant, increases a lot in numbers, and they become dominant in the community, and this is interesting. And bacteria are reduced, but they don't disappear. And viruses, and this will be the focus on the talk, go very high in numbers. Okay, you may think that they go very high in numbers because the seawater gets concentrated along the process, and therefore viruses also get concentrated. No, this is a succession of different communities along the gradient. The viruses that you have at the end, or the prokaryotes that you have at the end, are not the ones that you have at the beginning, mainly. And which is interesting, protists, well, I wouldn't say they disappear completely, but they disappear to a great extent. And so this predation exert on archaea bacteria disappears. So viruses become the main factor of biological control, okay? And now, just so who is living there? Which microbes we can find in the highest part of the salinity rank? Okay, mainly what we have are archaea, as I said, and archaeas are very frequently the main component is this square archaeon, which is called haloquadratum by obvious reasons. Wasby, because it was the name of the researchers who first saw this organism, and actually he called it the square bacteria. Actually, they are archaea, they are not bacteria. You see, they have these gas vacuoles here, and these have these like tortilla chips. When you shape, it doesn't look very biological, but it's a cell, a living cell. And also, and I will insist about the nanohaloarchaea. I will go now with this. And also within the bacteria, there is the main component is Salinibacter ruber, okay, which was the first bacterium that was extremely halophilic bacterium found to be ecologically relevant. And so we have this haloquadratum, the nanohaloarchaea, and Salinibacter, but Salinibacter is normally in way lower numbers. The nanohaloarchaea are really relevant, and they are even relevant in the field of astrobiology, because they have been found in one of the most extreme places, and several very extreme places in the world, which combine very high salt or very low pH, heterotrophic salts, and in other cases low temperature. So they are like one of the champions of extremophilism within the halophilic organisms. The nanohaloarchaea, this is like the common name, nanohaloarchaea, and it's not the taxonomic name. They were discovered by a couple of groups at the same time a few years ago. It was you have the year here. It's 2012, around that, and they are called candidatus, but because they haven't been brought into pure culture, but they have been enriched. They are very small. They are this part of this ultra microarchaea, like, you know, there are also these ultra micro bacteria, very small organisms. They can make up to 15% of the community. This is a lot. And they are very ubiquitous in hypersaline systems, okay? And there is an interesting trait of these organisms is that they are symbionts. I mean, either mutualistic or parasitic, but they need a host to live. Okay, the host is normally a haloarchaea, like the haloquadratum, not haloquadratum specifically, but haloarchaea from this phylum. These nanohaloarchaea are from the archaea, but the phylum, which was called DPANN, because of the groups that were included in this superphylum. This has changed now. There are more groups, but they are apart from the extremely halophilic archaea, as this haloquadratum said, others, which are here down in the Euryachaeaota. So they are from a different phylum within the archaea, okay? And they are parasitic, or well, they are symbiotic, they are widespread, and they live in they can live in places which are extremely tough. So what we have, when we look, imagine that we could make a composition of all the images. Here is what we see when we get water from one of these ponds, the crystallizer. And of course, we don't see this all at the same time. We can see the viral particles, we can see the cells, and here you have pictures that we take with the electron microscope. The cells can be rods, probably this is Salinibacter, then we have the square cells, and then also we have something smaller that are probably the nanohaloarchaea, okay? And in these waters, we have a lot of viruses. Viruses, I found this amazing the first time I learned this, that viruses can be observed under the light microscope. They need to be stained with a dye which is called cyber gold, cyber green, which stains nucleic acid and is very shiny. So you can see even particles that are below the resolution limit of the optical microscope. So when you stain with this cyber gold, you see the little dots, which are the viruses. Okay, here you see, for instance, when you get on the left water from the sea, from the beginning, and then you get water from the end of this gradient that I show you, the crystallizer, you see here many dots. Those dots are the viruses. So you can use this staining for the smaller dots. The larger ones are cells and the very tiny ones are the viruses. You can use this to count how many viruses, actually you don't call them viruses, I will explain you now, and have in how many viruses are present in a sample. So doing this kind of analysis, these are data from previous works from my group and also from other groups, and they are cited there. You can count how many viruses you have per milliliter of water, of this salty water. And we don't say, I mean, we don't dare to say, do we have so many viruses? We call them virus-like particles because, I mean, they look like viruses, but they could be something else. There could be vesicles, there could be gene transfer agents. So it's to be on the safe side, we call them virus-like particles. Sometimes we have observed them under the electron microscope and count them in this way. So in this way, it's like more clear. But in any case, these are the numbers. This is that we get when we count virus-like particles in these hypersaline systems. And you see that the numbers are huge, okay? And also, if you have we have the ratio between virus-like particles to cells can be up to 300. This is a lot. Normally, for instance, in seawater, it's like 10 viruses per cell. But in hypersaline environments, we have extremely high concentration of virus-like particles. In fact, those are the systems which harbor the highest concentration of virus-like particles that have been detected in aquatic natural environments. We can also see different morphologies. Remember that these systems, I was giving you the example of the crystallizer ponds of the solar salter, but in general, hypersaline environments harbor a rich community of archaea, and viruses from archaea have morphologies that are not present in viruses from bacteria. They also share some morphology, viruses from archaea and bacteria, but besides, there are some specific morphologies. Here you have, I will show you only some pictures. You have, this is the square archaea. You see that there are some viruses on top of it. And this picture, this is from a salty lake in the center of Spain. It has this gypsum crust, and you see here the diversity of morphologies. And here we also have pictures of viruses from salters and salt lakes in Argentina and Turkey. You see this like bullet and this lemon and filaments and different things. Okay, there is a quite rich diversity of morphologies besides this very high number of viruses. But now, please, I'm going to make just a parenthesis just to talk for a few minutes on the relevance of viruses of the biosphere. I'm sure many of you already are familiar with this concept, but in any case, I think it's quite interesting. Viruses infect basically all the different taxa that we know. I mean, very rarely there is one cellular organism that does not have a virus associated. Of course, you know that some viruses infect humans. We recently have this pandemic, so we are very aware of this. But most viruses in nature, I mean, the huge majority, infect microbes because most of the living beings in nature are microbes. And so viruses infect bacteria, infect archaea, and of course, they infect eukaryotes. So as I said, all cellular, or most, the huge majority of cellular organisms have their viruses, okay? And they are not always pathogenic to them. Some viruses can even be of help to their hosts. In terms of the biomass, this is the typical examples that they provide when you talk about the biomass of viruses let me check in the chat, because maybe there is some, I don't know if there is something. Yeah. I don't know. I have a question. Have you ever observed the viruses moving? No, because they are stained, and to stain them, you fix them. So you could also only show Brownian movement. I mean, but in any case, they are fixed and they are on a filter, so there is no way to stain them. Okay, so as I said, normally when you teach about viruses and so you provide these numbers, the biomass and the length. If you will be able to get all viruses on the planet together and weigh that, they will weigh as much as 75 million of blue whales, which is a lot. But if you see here at the left corner and down, you see this very on top of this square thing on the left top of this square thing, this is the biomass of viruses compared to the rest of the components of the biomass, in the oceans. I mean, they are still a lot, they weigh a lot, but compared to, I don't know, bacteria, they are not that heavy. But this example is providing some insight on how important they are. But the most important thing of viruses is the three main effects they have on and on nature. And you have this schematic representation here on top. Viruses infect the cells, their hosts, and when they infect their host, they different viruses may have different ways. Either they just reproduce inside the host and lyse them, they kill them. They can also and this is very common for eukaryotic viruses, become chronic. So they reproduce inside the host cell without killing them, and the progeny of the viruses is released slowly. They can also integrate the genome inside the host genome, and they can stay there forever. We have viral sequences in our genome, some viruses that sometime in our history become integrated in our genome. So by doing this, and because viruses are very specific, the viruses control the numbers and the diversity of their hosts. Since their hosts are involved in different parts of the trophic webs, viruses have an effect on the biogeochemical cycles because they can target specific components of the trophic webs. Besides, viruses can carry genes that are called auxiliary metabolic genes that can affect the metabolisms of the host. And viruses are like one of the main sources of gene invention. They have a very relevant role in lateral gene transfer and in inducing changes in the host genome. So they manipulate the host genome. They are like vectors of evolution. And besides, viruses change very fast. So this makes them very good models also to study evolution. So these are good reasons to study viruses, but why study viruses in salters? Besides these reasons, there are some others. For instance, salters are very, or hypersaline environments are very rich in archaea. And this is a very unexplored component of the viral diversity. In terms, for instance, of isolation, I will talk about this later. The viruses of Archaea are only a few, it's 100, a bit more. I mean, it's compared to thousands of viruses of bacteria. Besides, as I said at the beginning, there are viruses of DNA and RNA of single and double strain, but so far, there are no viruses of RNA infecting Archaea. This is very difficult to understand. So maybe it's not that there are not such viruses, but we couldn't find them. Then another trait of the communities in solar salters that very relevant to the study of viruses, as I said, that in solar saltern, the micro diversity, the overall diversity is not high, but the intraspecific diversity is very high. And this, together with the fact that viruses are the main control, biological control, or seem to be the main biological control, and make think that maybe this microdiversity is a way to it's a way of defense against virus predation, because viruses distinguish normally between different strains within a species. And another reason is that these systems are natural, but still control. I mean, you can make like in situ experimental evolution studies. You know, the experimental evolution studies are carried out in flasks. And then you have the like nature, the ocean, the soil. But this is something in the middle, that it's a pond that it's closed and you can manipulate and you can follow. And there is a natural community there. So it's good system, but still it's complicated enough. So the solar salters are a model to study the units of diversity that matter and ecosystem functioning. And this should also be true for viruses. And how do we study viruses in nature? Well, the traditional approach was to culture them. There is still true. I mean, it's not that it's forgotten. You culture them, you get the sample, you have a host. For instance, you want to culture a virus against a Salinibacter, this bacterium. You get a culture of Salinibacter, you get the viruses from the salter, you mix them, and you have techniques to observe that viruses are killing the cells. So afterwards, you isolate these viruses. This only works when first you have the host in culture and the virus can be cultured too. So it's not always very successful. And you may end up culturing something that is not relevant in the nature, or maybe you are lucky enough, but it's not very high throughput, and you may be missing something very clear, very key, because the hosts are not in culture. The most So, I mean, at this moment, one of the most used approaches is the metagenomics. And this is, well, you have a representation here. You have a community of cells and viruses. You see the square Archaea, Salinibacter, and then you, by doing a purification, you get the viral fraction. After getting the viral fraction, you extract the DNA sequence, and then by assembly and you recompose the viral genome. But you have to take into account where the viruses are, because viruses are in two places actually. When you have, imagine a natural community, microbial community, you have the cells, and if these cells are infected and are producing lytic viruses, there is lysis of the host and viruses are produced, you will have viruses inside the cells and outside the cell. You may have viruses only inside the cells because some viruses will have integrated the genomes on their hosts, or you may have viruses outside that are not infected. So you have to make sure where you want to look at the viruses. The best thing is to get viruses inside and viruses outside the cells. Okay, but this is like the technical design. And then you can also use single cell approaches. You can get one individual cell and sequence all which is inside. And if you have viruses, you will sequence the cell, you will sequence the cell and the virus. Or even you can do single virus genomes, which is pick up by cytometry one virus, one individual virus, one particle, amplify the genome and sequence. So I'm going to tell you a few examples of things that we have done to study the viruses and hypersaline environment using these techniques, and some conclusions that we may able to reach. We with my colleague Ramon Rosselló Mora, who was leading this study, we did and several researchers from around the world, which you have here. We were doing what we call the halophile sampling project. Okay, so we got samples. Actually, we got more samples than this, but these are the ones that I will talk about. We got samples from places all around the world, and from hypersaline environments, either coastal salters of marine origin or inland salt lakes. And from these systems, we, on one side, we have the cells and did the cellular metagenome, and have the extracellular viruses and did the viral metagenome. And one thing this is being analyzed by Borja Aldeguer-Riquelme, and the first thing we could see by just analyzing the sequences from these hypersaline systems, which are here in green, compared to other available viral metagenomes from all the world, and we could see that the viruses, the viral communities from hypersaline environments have a kind of hypersalineness. I mean, they are closer to each other in spite of the wide geographical distance. Okay, so you see, for instance, that soil communities in this purple color are very different to each other. The closer the dots, the more similar the communities. Marine waters are very close, and also fresh water are quite different, but hypersaline viruses are more homogeneous among them. And this is also, as we will discuss, related to how viruses go from one part to another. Let me check. So here you have the main hosts that, well, you have here the size of the viruses. We could see all over the world, whether the GC content that also tells you whether they infect hosts with high GC contents, whether they are very widespread or not, and which are the main hosts that can be associated to the more abundant viruses in the hypersaline waters in the world. And this is the organisms that I told you before, the Aloquadratum, nanohaloarchaea, Salinibacter, and also Alorubrum, which is an extreme halophilic. And also one thing we saw that there was quite interesting is that there is a very huge difference between the communities, the viral communities from inland systems compared to the marine systems, and that marine, coastal system, the solar salters in coastal environments harbor communities of viruses which are way more similar to each other than to that of the viruses in inland salters. So it seems that the seawater, and we have some data pointing to this aspect, the marine currents are playing a role on the distribution of hypersaline viruses, and of course, hypersaline microbes in general. This is not new, but because some years ago, some people isolated extremely halophilic bacteria from seawater. But this is a proof, well, or a clue that this is happening now, that the microbes, halophilic microbes, are being distributed in the world through the marine currents. Then we also have this, we focus on the study of viruses infecting the nanohaloarchaea. Why is this relevant? Because as I told you, the nanohaloarchaea are very abundant, very widely distributed, and they are symbiotes. And it's, at least for us, quite difficult to understand how the viral infection will proceed, because you have the host, the haloarchaea, I told you, then the nanohaloarchaea is a symbiont of the big one, and then viruses are infecting this nanohaloarchaea. So we have this three-side interaction: the large host, the nano, and the virus. But these nanohaloarchaea are not able to live by themselves. They need their host, they need the nutrients from the hosts, and they are vital components. So how virus infection proceeds is a mystery for us. So we aim at studying the viruses infecting nanohaloarchaea. And for this, we did a combination of cloning the viral genomes in phosmis, so getting the complete viral genomes from the viral components of the sample in a crystallizer, and then separating the cells on their side. So we then hybridize the viral genomes against the cells. Okay, so when one cell has hybridized against a viral genome, it means that this cell is being infected by this virus. So in this way, we were able to find nanohaloarchaea which were infected by viruses. So we had a lot of 45 nanohaloarchaea single amplified genome. When I say separating the cells, here I mean the individual cells, okay? So we had like 45 different nanohaloarchaea. We sequenced them, and we found that some of them were infected by viruses. So we went on and studied these viruses. There were a total of 14 viruses that we had. We had the cell, remember, we have picked up the cell with the single genome techniques, sequence everything inside, and we got the nanohaloarchaea genome and the viral genome because this cell was being infected. So we then analyzed the viral genomes and saw that, in fact, they were real viral genomes with hallmarks of head and tail viruses. Okay, so what we saw is like 1/4 or 1/3 of all the nanohaloarchaea cells that we were able to detect were infected by at least one virus. And when we check whether these viruses were widespread, we saw that when we got, you have here different bonds of different concentration, and the names are here. Anyway, here what you have to look is, on the left is the names of these new viruses we found within the nanohaloarchaea. On top here, the brown things, the darker, the higher salt. And these small squares is the abundance. When you have red is very abundant, and then you have the scale here. So you see that some of these viruses on the left are very abundant because they are red. For instance, the first line, it's you have this virus which is abundant, but as you see, it's very abundant when these colors are darker. The brown color is darker, that means higher salt. So what we saw is that these viruses are present there, but they like the high salt systems, the highest salt bonds, which is normal because the hosts also do the same. And when we went to the halophilic sample, halophilic project that I told you before, we also saw that these viruses that we have detected in our samples from Alicante, which was where we got all of them, were also present. Here on the left is the name of the viruses, and on the right is different names of salters we have analyzed. We see that when you have the dot, is that the virus is present, and the higher and dark, and you have the scale here, it's more abundant, okay? So we see that these viruses are quite abundant, and they are present both in the intracellular fraction, so in the cells, and in the extracellular fraction. So what does this mean? For instance, let's look at the second line here. You see dots in this is, let me put my glasses on. You see here A and R, right? Same sample NAR. You see this dot here, the second line, but if you go to the extracellular fraction, you don't see anything, right? When you have this, the second virus, you go metagenome, you see a lot of dots, and then you go to metavirome, you don't see anything. What does this mean? This means that these viruses are inside the cells, but not outside the cells. Okay, so this can be for different reasons, and this takes us to a different thing that we discovered using these systems. When you have the viruses inside but not outside, this may mean that the virus has integrated the genome in that of its hosts, right? But normally, when viruses integrate the genome in their hosts, these viruses have some hallmark genes. But we saw something that was a bit strange. We went from the laboratory to, from the environment now to the laboratory. So the explanation of viruses being inside, but not outside, means it's that viruses are lysogenic, what I told you before. They integrate their genomes. But we saw also that when the viral concentration is very high outside, some viruses go to something that is called pseudo-lysogenic, okay? It means that these viruses remain inside the cells, they don't kill the cells, they don't integrate into the genome, and then they are not transferred symmetrically. So this is a way of maintaining a very high virus population, as we have in solar salters, without killing the cells. So viruses go inside, but do nothing, basically, okay? And how many viruses go inside the cells depend on how many viruses are outside, but this solves this mystery we have of this very high ratio of cells, of viruses to cells in Hypersaline environment. And just the last example to finish something which is less target, but I think it's quite interesting, but this is like more questions than answer, but it's a way of how hypersaline environments can be used or to monitor viral evolution online. I mean, and in continuous, you can watch viral evolution. This is not only in hypersaline environments, but in this case, of course, it is. Okay, so this system is quite interesting. This is a very ancient salt turn. It's called the Salt Valley, and it's in the north of Spain, and it's constituted. You have here on the right. It's a salt diapir. I mean, it's a salt rock that gets dissolved and goes through a spring. You have here La Ontana. This is the spring. Goes up to the surface. And when it goes up to the surface, well, this water is used to fill some ponds, and it's used to collect salt. The Romans, more than 2,000 years ago, also already used these salters for salt harvesting. Anyway, the question is that we have, you see, the important thing here is you have this salt diapir, which is underground. And there is thermal water that goes and dissolves the salt. And the salt goes up, and this goes up through these three wells you see here. You have this lantana, which is just the spring water. The salty water is like 20%, it's quite salty. And there is a puddle here. I mean, just you see the picture, it's just a small puddle. And then we have these two springs that have a reservoir. You see, these square things are reservoir. So you have the water that comes from the underground, and here on the left, that goes out all at once without waiting. But in these ponds, in these systems, water has to wait for some time, not much, because it's quite fast. So we did an analysis of all these four systems, which is the spring water, this puddle, in these two reservoirs. Okay, the colors are always the same. Red is like the underground water, and this is the one that has been waiting for a while, and then these two ponds, these two reservoirs, and yellow and green. So you have here, we're counting bacteria and archaea by fish and doing viruses, and also the diversity. Here you only have to look at the colors. These are the numbers on the x-axis is just different species. In red, you have all these species, but you see that, and on the y-axis is the abundance. Okay, you see the profile of the species in the spring water, and you see that some species disappear when they go out, and you get, for instance, and on the right on the distribution, species that appear only in certain points. Okay, but the highest diversity it's in the spring water. The upwelling is the highest diversity. This comes from the underground water, the salt, and everything. And here, what I told you, the evolution. So we did the viral metagenomes and the viruses and the cells. So, and we reconstructed the different genomes. Okay, so let's look at the red, brownish thing. A, B, C, D mean different genomes. We were able to reconstruct the genomes by sequences. And we were looking for these genomes in the sequences that we got from the viruses. And of course, they were present there. And if we go to the cells, we saw that most of these viruses that were outside the viral metagenome were not inside, okay? So only one, this H virus could be found in the cell metagenomes and the viral, okay? So this type of diagram is when you have something below, under the name of the genome, means that they are present there. If you have this line, it means that the whole genome is present, okay? So we saw these viruses that were not inside the cell metagenomes, but the very same viruses, when the water went from the upwelling, the red, to the paddle to the yellow, you see that some of these viruses start to be inside. They are on the right. They are not only on the virus side, but on the cell side. That means that these viruses have started to infect. And this goes very fast. Those viruses that were doing nothing on the spring water, when they come out, they start infecting. And we can also see that you compare the viral genome from the spring, okay? You have, this is the different genes on top. You see that this virus is not exactly, you see here some areas that are missing. This is the genome, and you see some areas that are missing. What does it mean? That these areas of the genome have changed. We have catch the evolution in action, because we were able to see how a virus that had a certain genome in the spring water had changed, or was a selection of some small variants in a few, in a very little time. So, and with this, I'm going to finish. And as I said, the solar salters can be considered models to study the units of bacterial diversity that matter for ecosystem functioning, and I hope I have convinced you that it also includes virus-host interaction. And this is part of the people who have been working with this, and which I'm very proud of, my group, and I thank you very much for your attention, and muchas gracias. And I'm going to share, so in case you have any question. I will answer. Armando Thua, I haven't found Archaea in these samples. We found in other samples from underground water, but not in this here. And we the sediment sample, yes, we have looked at sediment samples, yes, but the problem with the sediments is that purifying viruses, the viral fraction is more difficult, but we are working with marine sediment, and yes, we are working. But the interest of the salter were the Archaea and the biological control. Sediments are very complicated systems, but yes, we are working with these samples.
Any other questions before we finish up?
Anyway, if you have any questions, you can send me an email. I hope you understood, because I was, I mean, not because I understood, it was clear, because maybe you couldn't see the arrow, and that's my email. Yes, sure. It's very easy. Here you go, my email.
Excellent. Thank you so much. We will finish up there for today then. Thanks to everybody who's attended as well. Bye.
Bye-bye. Thank you very much.